In the previous video, I talked about a depolarization and repolarization. This is what produces an action potential. Now, only certain cells can do this. These are neurons and skeletal muscle fibers. They are considered to have excitable membranes, and that depolarization, repolarization excites the membrane and produces that action potential. This is a method of information. This action potential is spread down the plasma membrane very, very quickly in one direction. Which of the following contain gated calcium channels to activate contraction? I don't think I mentioned that in the video. You'd get that from your reading. Terminal cisternae. In this chapter, we're going to be learning about a neuromuscular junction. This is the connection between the nerve and the muscle cell. Every time a muscle cell contracts, it must get the information to contract from a neuron. So this junction is where a motor neuron controls a single muscle cell. Now the neuron can branch and control multiple muscle fibers. However, every single fiber must be stimulated by a neuron. So we're going to be learning about this neuromuscular junction. There are quite a few terms here. There's an excellent video in Moodle. I would watch that multiple times and possibly fill out the worksheet that's available there in Moodle as well. So at the neuromuscular junction, it is where the motor neuron, comes down to the muscle cell or fiber. The end of the neuron has, let's see if it shows. It doesn't show it. Oh, here we go. So here's our motor neuron coming down to the muscle fiber, and the end of the neuron has what's called an axon terminal. Inside the axon terminal will be vesicles containing neurotransmitters. So here is the neuron coming down to the muscle fiber. Here are the vesicles, and they contain a neurotransmitter called acetylcholine. You've got to learn that transmitter. That is the only transmitter at the neuromuscular junction. Once we get to the nerve-to-nerve junctions, you'll see a variety of neurotransmitters, but in the neuromuscular junction, there is only acetylcholine. So these red dots represent the acetylcholine. Now what happens is the muscle fiber actually kind of indents right where the nerve ending is going to come, and that's called the motor end plate. So over here on the left, you can see the motor end plate. There is a gap there. The neuron does not actually touch the muscle fiber. That gap is called a synaptic cleft. Here are junctional folds in that motor end plate. And the acetylcholine will have to be released and cross the synaptic cleft and bind to acetylcholine receptors on the motor end plate. So again, here's our motor end plate. It has infoldings called junctional folds, and this is where we're going to have acetylcholine receptors. You will also notice this yellow moon here. That represents acetylcholinesterase, which will break down the neurotransmitter acetylcholine. The synaptic cleft is the space between the neuron and the muscle. So an action potential comes down the neuron, arrives at the axon terminal, and there is a change in the membrane permeability of the axon terminal, and this causes acetylcholine vesicles to fuse with the neuron and release the acetylcholine by exocytosis into the synaptic cleft. So here you can see the acetylcholine is in the vesicles, and here exocytosis is releasing the acetylcholine into this gap. The acetylcholine has to diffuse across the gap and be received by these receptors. Once the acetylcholine is bound to the receptor, the sarcolemma, which remember is the muscle membrane, the muscle fiber membrane, the sarcolemma changes its sodium permeability and sodium rushes in to the muscle fiber. When the sodium rushes into the muscle fiber, it generates an action potential in the sarcolemma. Now, the acetylcholine has to be removed from the receptors so that those receptors can close, and the acetylcholine is removed by the enzyme acetylcholinesterase. The action potential is generated at the motor end plate. It spreads across the entire sarcolemma. The action potential moves down the sarcolemma and into the T-tubules. So the action potential travels here. And is going to it's like it enters the entire muscle cell instead of just being on the membrane by traveling down the T-tubules. So it travels down to the T-tubules to the terminal cisternae of the sarcoplasmic reticulum and changes the permeability of this organelle, this sarcoplasmic reticulum. I hope you remember what is stored in the sarcoplasmic reticulum. You got it, calcium. So once the action potential travels down to the sarcoplasmic reticulum releases the calcium, and this begins the excitation-contraction coupling. That is, the action potential is coupled with contraction, that sliding filament that will shorten the sarcomeres. Which of these is the part of the muscle fiber stimulated by the motor neuron? Motor end plate. What chemical is secreted by motor neurons at the neuromuscular junctions in skeletal muscles? Acetylcholine. Which of the following ends the stimulation of a muscle fiber? ACHE represents acetylcholinesterase, which is an enzyme that breaks down the acetylcholine. So ATP is required in this whole business here. And we're gonna see exactly what happens when the muscle fiber contracts and what happens to that sarcomere. So here we see resting sarcomere. The myosin heads are energized and cocked, and this requires ATP. When the calcium cycle is the contraction cycle begins when calcium is released from the sarcoplasmic reticulum. Troponin has a binding site for calcium. So we're going to see that calcium's important in moving troponin. So once calcium binds to troponin, troponin moves. And as it moves, it's going to move that secondary protein that we're interested in, the tropomyosin. The tropomyosin was covering up a binding site on the actin. So calcium enters, it moves the troponin, which then moves the tropomyosin, and it opens up the binding site on the actin. Once the binding site is opened up, then the myosin head here, this purple head, can bind to the active site on the actin. This is called forming a cross bridge. You've got millions of these myosin heads binding to the actin filaments. Once the myosin heads are bound, the myosin head pivots And you can imagine if this head is pivoting to the left, it's pulling that actin filament to the left as well. This is called the power stroke. And once this occurs, the ADP and phosphate are released. The cross bridges detach, and a new ATP attaches to each myosin head, and the myosin releases. So ATP is needed for the power stroke, and ATP is needed to release the myosin head from the actin. The myosin will be reactivated. The free myosin head splits ATP into ADP and phosphate, and the released energy is used to recock the myosin head. And you can watch this video. The cycle will repeat while calcium is high and there's enough ATP. The calcium levels will stay high only if action potentials continue. When the stimulus ends, the sarcoplasmic reticulum calcium channels close, and there are particular calcium ion pumps that are going to return the calcium from the cytosol back to the sarcoplasmic reticulum. And the troponin-tropomyosin complex resumes original position. Remember, the tropomyosin was covering an active site, and the troponin was kind of holding it in place.